If you want to know a fish's age, just take one of its scales and count the number of bands spreading out from its center. For trees, you can count their rings. For narwhals, their tusk layers. And for blue whales, their layers of earwax. When it comes to humans, scientists still have not found any visible signs that show our age with anywhere close to the same precision. But in the past few decades, they have found small, invisible markers hidden inside the body that do change over time. And they may hold even more information about our health, history, and future. The first of these markers was found in the 1990s. Telomeres are repeating sequences at the ends of DNA strands that protect chromosomes from joining together. However, each time a cell divides, part of the telomere gets cut off. These small losses can build up over time, and once telomeres get too short, cells lose their ability to copy themselves and eventually die. This slow shrinking process led scientists to see telomere length as a promising, though imperfect, marker of aging. As technology improved, scientists learned how to detect even smaller changes to DNA, like the addition of individual molecular tags called methyl groups. This DNA methylation can control gene expression, including turning genes on and off. It can happen quickly and often, allowing cells to respond to different situations and carry out normal functions. But researchers noticed that at certain places in the genome, methylation levels tend to change more slowly and predictably as we age. The reason for this is still unknown, but these changes may be linked to stress or caused by random mistakes during DNA replication. By measuring DNA methylation patterns from hundreds or thousands of these places, scientists have developed what they call epigenetic clocks as another way to measure aging. A third way our bodies change over time is through inflammation. Proteins that are important parts of the immune system usually increase when we get sick, then go down again once our body has recovered. But as we get older, their usual levels rise, leading to chronic low-level inflammation. This process is called inflammaging. Scientists are still not sure why it happens, but it could be due to repeated contact with pathogens and other stressors. Another explanation could be the process of cellular senescence. When cells get old and stop dividing, they release inflammatory proteins. And by measuring the level of these proteins, scientists have developed yet another clock that can accurately measure aging. While all three of these markers are great additions to the age-estimating toolbox, they do not perfectly match chronological age. Instead, they are best at estimating something called biological age. Biological age is a measure of how well your body is working, how much damage your cells and organs have built up, and your overall risk for certain health problems. Unlike chronological clocks, biological clocks tick at different rates for different people because of genetic differences, as well as health, stress, and many other environmental factors. And different tissues and organs inside the body may even biologically age at different speeds. In many ways, these clocks are still a work in progress. For example, most experts advise against personalized biological age testing. While these tests are sold commercially, the value and accuracy of these measurements are still unclear. Right now, these tools work best at the population level, when data from many people are studied. However, many scientists hope to change this in the future, since accurately tracking this information could revolutionize personalized healthcare. For example, one day a person's faster biological age might help doctors detect an unknown health condition much earlier. And other researchers are looking for ways to slow down, or even turn back, these biological clocks. Though so far, most attempts to do this have been largely unsuccessful. Besides, researchers still do not know if telomeres, inflammation, or epigenetics are actually causing aging, or if they are simply neutral markers of other processes happening in the body. We may be learning to read the language of aging, but we still have a long way to go in understanding its grammar.